histochemistry to come at least close to the diagnosis of whether it is gliomas, adult, pediatric, or embryonal. We have actually chosen these three major topics because these cover at least 75% of the tumors that you would see. Of course, we have a very large chunk meningiomas, but as Dr. Vani mentioned, molecular is possibly not one of the many approaches for meningioma. So if we take tumors with a lot of molecular approach, these three would cover the larger segment of the CNS tumors, and they have been covered quite extensively by all the three speakers. Now we come to the last speaker for today's CME, Dr. Vandita. She is the host, so thank you very much for giving us this opportunity. And she would be presenting the molecular platforms in the next 15 minutes. So over to Vandita.
Molecular techniques in brain tumor diagnostics as briefly as I can. So there has been an exponential growth in identification of different molecular aberrations in CNS tumors, and also the techniques that are available to diagnose them. And therefore, I'm only going to cover the most important aberrations and the important techniques that are used. So I would outline my lecture as beginning with a rationale for molecular diagnostics, the spectrum of molecular aberrations and the recommended techniques to look for them. I would be comparing the molecular assays with respect to their turnaround time, cost, sensitivity, and recommendation level, principles of common tests, algorithms for performing the tests, and conclude. So WHO 2016 and 2021 have suggested incorporation of molecular markers because they give additional diagnostic and prognostic information, which histology cannot. Histology is marked by inter-observer variation, and molecular diagnostics, on the other hand, can also differentiate histologically similar tumors into prognostic and predictive groups, which is important to molecularly stratify patient cohorts for trials. So this is a rationale for molecular diagnostics in CNS. The spectrum of genetic aberrations that are available in CNS tumors encompass promoter hypermethylation, copy number variations, fusions, deletions, translocations. And I would only look at the important ones according to the latest NCCN guidelines. So IDH1 bar 2 mutations, these are required, assessment of these are required for diagnosis, and the detection methods used are immunohistochemistry and sequencing, which can be Sanger, pyrosequencing, or NGS. 1p19q co-deletions, as we know, are essential for the diagnosis of oligodendrogliomas, and the methods commonly used to detect them are FISH, PCR, SNP, and NGS. FISH is no longer the recommended method, having been replaced by SNP analysis. MGMT promoter methylation are essential for diagnosis of WHO grade 3 and 4 gliomas, and the methods used to detect them are methylation-specific PCR, which can be quantitative, high-resolution melting analysis, and pyrosequencing. ATRX mutation is recommended, and the methods to detect it are IHC and sequencing. TERT promoter methylation by sequencing of the promoter region. H3F3A mutations have to be looked for in the appropriate clinical context, and the gold standard to look for these is sequencing because it detects all the mutations of histone, including G34 and H3K27M. IHC can serve as a surrogate, but in ependymomas, methylation profiling is a preferred method. Now, BRAF mutations can be fusion or BRAF V600E mutations. For BRAF V600E mutation, sequencing is a recommended method. For fusion analysis, RNA sequencing, PCR-based breakpoint methods are used. FISH is no longer considered reliable for this. In ependymomas, the important alterations looked for are RELA fusion and MYC amplification, and the methods to detect them are RNA sequencing and FISH, apart from methylation profiling for stratifying these ependymomas. Medulloblastomas, as already commented by Dr. Shilpa, there are 4 molecular groups in it, and the methods to detect or classify them into each of these groups are starting with IHC, followed by expression arrays and DNA methylation arrays. So all of these molecular assays can be simple or complex, or they can be specific to the epigenetic alteration we are looking for, most commonly methylation. For methylation, the techniques which are used are methylation-specific PCR or quantitative PCR or MLPA or pyrosequencing or methylation profiling. Based on the method of the techniques used, they can be classified into 7 types. The first is cytogenetics in fish, chromosomal microarray, which includes comparative genomic hybridization or SNP arrays, amplification methods, all based on PCR, DNA high resolution melting curve analysis, sequencing methods, next generation sequencing, and DNA methylation analysis. If we were to compare the turnaround time for all of these molecular assays, we can see from this figure that the highest time is taken by methylation profiling, SNP arrays, and NGS, because there are multiple steps involved in doing these assays. Although they are limited by this, however, the amount of information that comes after doing this is much more than the simpler assays. And with time, as these techniques become more economical, they will replace the simpler assays. If we were to compare the cost, sensitivity, and molecular abnormalities which are assessed by each assay, the cost per assay is highest for whole genome sequencing and whole exome sequencing and methylation profiling, precisely because there are multiple steps and also a lot of data which needs to be analyzed. The best sensitive method remains NGS. In order to look for point mutations like IDH1 or 2, ATRX or TP53, next generation sequencing is the best method. In order to look for copy number variations, code deletions, and copy number variations like EGFR amplification, SNP array is the best method. In order to look for translocations and fusions, FISH still has a role to play. And in order to look for methylation, methylation profiling, and methylation specific MLPA are the best methods recommended. So if we look at each assay in short, we know that fluorescent in situ hybridization is a technique to visualize the presence of a DNA target in its natural position in the chromosome using a fluorescent tagged probe, which on hybridizing releases fluorescence, which can be visualized under the microscope. Now, FISH is only limited to known targets. And depending on the probes, that is numerical chromosome enumeration probe, dual color, dual fusion probes, or break apart probes, you can look for presence or absence of chromosomes, or translocations where both the partners of the translocation are known, or break apart probes where only one partner is known. The specific cutoffs for calling a FISH test as positive or negative have to be established during probe validation in each laboratory. Now, this is an example of a FISH dual color dual fusion probe, where you can see in this figure that there are 4 signals. Red and green are separated, and there are 2 which are fusion signals. So having 2 fusion signals is specific for a fusion. So FISH is advantageous because it is more, it has higher resolution than conventional cytogenetics, can be done on FFP tissue, and it is relatively fast. However, there are limits to FISH as well. Its resolution is not good enough for smaller alterations. So for that, we have chromosomal microarrays, which include array comparative genomic hybridization and SNP array. These are cytogenetic methods to analyze genomic copy number variations, which are smaller than those that can be diagnosed by FISH. So prior knowledge of a chromosomal aberration is not required in these chromosomal microarrays. No culture is required, and therefore they are better than FISH. The types of chromosomal microarrays that are available are CGH and SNP. SNP is advantageous over CGH because it can detect not only copy number changes, but also loss of heterozygosity. Also, CGH requires a comparative control, which is not needed in SNP. So a comparative genomic hybridization, what happens is we have artificially prepared probes, which are either bacterial or oligonucleotide probes put on a platform. The test or sample DNA and control DNA labeled with different dyes is hybridized to this DNA chip, and signal intensity is measured of test DNA compared to control DNA. Higher the intensity indicates duplication, lower intensity indicates deletion. So these CGH is therefore used for copy number changes, losses, or gains. SNP is better than CGH because it can detect loss of heterozygosity. Because for a given SNP array assay, 2 alleles are seen. So you can make out if there is homozygosity or heterozygosity. Now, interpretation of the microarrays is that the interpretation is not positive and negative. Interpretation is categorized into 3 categories. Array can be called as pathogenic, or of uncertain clinical significance, or benign. And this is made after comparing the SNP detected with peer-reviewed publications. Now, this is an example of how SNP is better than FISH for 1p19q co-deletion. Now, the Figure marked A is an oligodendroglioma and B is a glioblastoma. As you can see in A, chromosome 1p and chromosome 19q, the red line is a normal neutral copy number. So anything below that detects deletion. So in oligodendroglioma, there is complete loss of chromosome 1p and chromosome 19q, whereas the SNP can detect a partial loss of 1p and 19q in a glioblastoma. Therefore, the resolution of SNP is favorable in these contexts. So genomic resolution of SNP arrays is better than FISH and conventional cytogenetics as it can detect submicroscopic genomic copy number variations not detected by other methods. Now, very important assays, DNA methylation assays. Now, DNA methylation is an epigenetic modification that regulates gene expression. Methyl groups are normally added to the cytosine nucleotide, which followed by guanosine at CPG islands. These CPG islands are rich in the promoter region of genes. Methylation at these sites causes decreased gene transcription and plays a role in human neoplasia. So significance of identifying or distinguishing methylated or from non-methylated DNA is that it predicts response to alkylating therapy in high-grade gliomas. There are different methods for assessing DNA methylation from simple to complex assays. All start with a bisulfite conversion method. So the principle of bisulfite conversion is that unmethylated cytosines convert to uracil, whereas methylated cytosine is not converted after bisulfite conversion. And then when we take a ratio of methylated versus unmethylated, we can quantify the amount of methylation. So after you get the product bisulfite conversion, you can detect the product by methylation specific PCR, sequencing, or melting curve analysis. Non-bisulfite dependent methods are also there, and the need for these is that bisulfite conversion is a difficult thing to do. So there are restriction, endonuclease digestion, and affinity enrichment methods that are available, but they are not very commonly used. Now coming to PCR. So before we start the methods based on sequencing, we start with PCR because it is the most simple sequencing platform available. PCR is based on repeated denaturation, annealing, and elongation over 35 repeated cycles, which gives rise to an amplicon, which can then be visualized by different methods. So the limitations of PCR are that if there is any contaminant in the mixture, that would also get amplified. Now, what are the variants of the PCR that are very relevant for CNS tumor diagnostics? There is quantitative PCR, the principle of which is that there is real-time measurement of the DNA accumulation via fluorescence-based approaches during early exponential phases of PCR. The computer constructs an amplification plot and is able to give us quantify the concentration of input target DNA sequence. Advantage of real-time PCR is that you can use it on any kind of sample, fresh or FFPE. It is a robust approach, but the disadvantages is that there is amplification bias. Now, there are lots of PCR variants available depending on what you're trying to do. What is the question you need answered? Multiplex PCR is, there are multiple primers are used for different loci within the same large gene. It is used to detect sequence at multiple sites, massively parallel sequencing, like in next generation. But it needs a lot of control. Nested PCR is a type of PCR in which you have 2 rounds of PCR. In the second round of PCR, the products of the first round serve as template. Now, this makes a PCR reaction very, very specific, but it is also very sensitive to contamination. An example of nested PCR is a methylation specific PCR. Now, multiplex ligation dependent probe amplification, or MLPA, is a variant of the PCR. The importance here is there are 2 adjacent DNA probes of different sizes which will hybridize to the target sequence. Why you need 2 probes is if there are large deletions, the commercially available probes are not enough to cover the entire deletion. So you can take 2 different probes. Once they hybridize to different parts of the target, they will ligase and fuse, and these can be amplified. So it is able to detect large deletions, copy number changes, and in order to make it methylation specific, you can do a bisulfite conversion step. Now, DNA high resolution melting curve analysis is another method to screen for sequence variations due to deletions or insertions. So high resolution melting curve analysis will only answer the question if a sequence variant is present or not, but it will not identify the sequence. It can be used to detect methylation as well. Shape of the melting curve will be different from a normal DNA. And melting curves of test DNA when compared to melting curves of normal DNA will help us know whether a variation is present or not. Sanger sequencing is the first step of all sequencing methods, the most simplest form of sequencing. We all know about it. It is based on chain termination strategy. So the basic thing is that each time Taq polymerase incorporates a DDNTP, chain synthesis stops. If this DDNTP is fluorescent labeled, then the fluorescence signal order of the peaks produced would correspond to the DNA sequence. So Sanger sequencing can detect even smaller alterations in the form of point mutations, small insertions, and deletions. Pyrosequencing is similar to Sanger sequencing, but instead of fluorescence, you have release of pyrophosphate when nucleotides are incorporated. Pyrophosphate produces ATP, which oxidizes luciferin and produces light. Intensity of the light is directly proportional to the nucleotide and its quantity. So this is an example of a methylation specific pyrosequencing. On the left side, what the graph shows you is unmethylated cytosine. Unmethylated cytosine is not converted into unmethylated cytosine is converted into thymine, and therefore cytosine peaks are absent in the pyrograph. Whereas methylated cytosine remains as it is, and therefore cytosine peaks are present before the G. So you have C and G, this is a CpG island, and in before the G, you have the C peak present here. Whereas on the unmethylated side, before G, you do not have the C peak. For example, here, if you can see my cursor. So when you do a ratio of methylated versus non-methylated amounts, you would know, you can quantify the methylation present. Now, the last type of sequencing analysis is massively parallel sequencing or next generation sequencing. Steps involved in NGS are several. Multiplex PCR amplification, DNA library preparation, clonal amplification, and sequence analysis. The two platforms that are commonly used are so the common platforms commonly used.
Vandita, you will have to possibly hurry up.
Yeah, Iron Torrent and Illumina. Ma'am, just a minute. And the applications for NGS are whole genome sequencing, exome sequencing, and targeted hotspot gene panels. So next generation sequencing has several steps that need specialized expertise and training. Lack of bioinformatics resources for analysis, instrumentation costs, and large amounts of data generated are limitations for using this. Methylation specific PCR and pyrosequencing, I have already explained. Basically, the results of these is expressed as a methylation index. When this index is less than 4, it means there is unmethylated DNA, 4 to 16 borderline, and more than 16 is methylated. Now, although there are so many assays, not all of them need to be done. Depending on the tumor type, you can make an algorithm, and so you can see the red line for, yeah.
Anyone is logged in the auditorium, kindly log out.
You can stop at the histological diagnosis, and only in a few cases these molecular assays are needed. So if we use these guidelines, for example, by ENO or other groups, then you don't have to spend a lot of money on these assays. So there are these algorithms which have been recommended for gliomas, adult and pediatric, as well as ependymomas. So the take-home messages would be that there is a need to develop institutional and regional algorithms to determine the sequential use of these molecular tools based on local resources and tumor epidemiology. The pros, cons, and limitations of each method have to be considered for each tumor type. Histology still remains the first rung cost-effective method, and molecular diagnostics do not replace it but are complementary to it. Thank you.
Thank you, Vandita. That was also an excellent overview of all the molecular platforms that can be used. So we come to the close of this session. If there are any, I do not see any questions in the chat box as yet. So if the speakers are there and would like to give any comments before I summarize thing. Vani, Geeta, Shilpa, Vandita, would you like to say anything before we close the session? Anything to highlight? Vani, are you around?
Mute.
Yeah,
I was trying to unmute.
Yeah.
I
Can
agree
you,
with,
would you like to say something?
Yeah, I think I agree with you that the session was very comprehensive. So we've had different aspects of CNS tumors, and what every speaker, I think, started with the older concept and moved on to the newer concept and the WHO 2021 classification. So I agree with you that it is predominantly the gliomas, the adult, the pediatric, the glioneuronal tumors, as well as embryonal tumors. That is where a lot of moleculars are going to come in. But what is very important is for the participants to know is that this is going to be, it's not completely a molecularly defined classification. It is definitely a hybrid classification. I think that is a point that you should remember, that it's not going to be molecularly defined for all the tumor groups. There will be some which are molecularly defined, but some will be hybrid and some will be based on only their histology. And moleculars will be used probably for their prognosis and better for their targeted therapy, better understanding. So these are some things that you should take home, because it's like any other evolving classification where you have histology, as like Vandita told us in the end, histology is the cornerstone. And without histology, you cannot ask for a molecular test. You should be very clear about what molecular test you want in that particular case based on histology. So I agree with you on that. And I should congratulate the other speakers, my co-speakers, who have done a very good job of all the other talks. Thank you, Chitra.
Geeta, are you there?
Yes, I am. Just to say that pediatric gliomas have really, compared to what we used to diagnose before, we've learned so much now with all the new genetic alterations that have been detected. But definitely histology is the cornerstone, and we just have to move in an algorithmic manner through it, at least to the point of narrowing down what it could be. And important points are things like in infants, even if you see a high-grade histology, they are going to behave as good tumors. And so one mustn't, think of a bad prognosis in such cases. So while histology does play a role, age, clinical presentation, location, everything comes into play, especially in the pediatric age group.
Thank you. Shilpa, Vandita, anything more before I summarize? I must agree that this was a fascinating session, and within the period of time allotted, a very good coverage of the entire new classification, which is coming in a few months' time. What I would like to tell the young participants is, again, do not shy away thinking it's all molecular. Histology is the cornerstone, as everybody has highlighted. From histology, the next algorithm would be immunohistochemistry. And as every speaker has highlighted, there are a lot of immunohistochemical markers, immunohistochemical surrogates of genetic markers, which have come in, like the H3K27M, the IDH1 antibody, which can be used, and immunohistochemistry is there practically in all pathology labs. You have to just increase the spectrum of antibodies that you use to include some of the CNS tumors also, at least for gliomas. ATRX can suffice for a large number of gliomas, and P53. So MIB1 is already there. So in this way, you can look at, for medulloblastomas, which is the commonest tumor, a malignant tumor in children, You can look at the beta-catenin, GAB1 and GAP1. So from histology, you move on to immunohistochemistry, and at least I would encourage most labs to include within their immunohistochemical panel, whatever they already have for lymphomas, lung cancers, and other oncology, some of the important neuro-oncology immunohistochemical markers. Then we go on to the next technique, which possibly can be introduced, would be the FISH. And FISH can be done on FFP, and I know it needs a larger infrastructure of microscopy, and the probes still are commercially available, are costly. But if at all, the next thing then go CNS tumor, which in help you, and then of course sequencing, etc. One more thing to add about the WHO 2021 classification is that they have introduced at the end of every chapter what is essential diagnostic criteria for every tumor and what is the desirable criteria. So I think this will be a very important addition in the WHO 2021, and that at the end of every tumor, you have the essential and the desirable diagnostic criteria, which will help us to come to a diagnosis. And last but not the least, yes, the new technique which everybody is talking about is the DNA methylation. And that is coming in even in the WHO 2021 as a means of diagnosis of some of the rare tumors. Some of the rare tumors in the classification have been recognized because of DNA methylation. And now there is a platform, a public platform by the German Neuro-Oncology Network, where if you can do DNA methylation and feed into this network, you get back a diagnosis in difficult cases. So things are moving in a lot of different directions, but in our resource limited settings, histology, immunohistochemistry, supplemented with some of the molecular markers, we could possibly just do a good justified diagnosis to most of the CNS tumors. Thank you all for this patient, to all the attendants. I saw that there was about 40 to 45 participants for this pre-conference CME. Thank you all for your attention. I must congratulate the director of the Institute. I know he's extremely dynamic for having given AIIMS Raipur Pathology Department to host this conference. And on behalf of all the speakers and I as the chairperson, thank the organizers for this wonderful opportunity. Thank you all once again. So I think we can conclude this session.
Thank you very much, madam, for giving the session. And I thank all the speakers also for their excellent lectures. Thank you. So now we move on to the next session, that is on lung tumors.